Olá, pessoal da Bocaina, eu sou o Lucas Perillo e estou aqui na Argentina, na unidade do Crilar, aqui em La Rioja, Anidiaco, que é o nome da cidade. E a gente está aqui há quase 10 dias já fazendo um curso de identificação de Hymenoptera, e um dos tutores, um dos professores é o Matt, e vou ter o prazer agora de conversar com ele aqui sobre algumas coisas. Matt, it's a huge pleasure to talk to you.
Thank you. Likewise.
And to start our interview, I'd like to ask you your history in biology career. Did you made the biology in graduation and master degree, PhD? Where did you get it?
So I got my undergraduate at UC Riverside, and at the time, my plan was to go to medical school.
Oh, really?
I got a job. I needed to get a job on campus. The only place hiring was entomology, and I wasn't aware of the science of entomology. I didn't know what the word meant. And as it happened to be, as life throws these very bizarre possibilities in your path, Riverside happens to be one of the centers of excellence of entomology in North America, if not the world. So it makes sense that they were hiring because they had a big department. And my job was to sort pitfall samples of terrestrial arthropods with a speciality in the Hymenoptera.
Right.
We're looking at conservation, it was a big project. Thousands of samples. Most of the Hymenoptera were ants. However, in the same samples were these tiny, tiny wasps that I thought were baby wasps. That's how naive I was. And I later found out these were parasitoid wasps, that they attacked eggs and larvae of other insects. They were just like a big wasp, except just shrinky, really small, and had some pretty spectacular life history traits. And in a few months, I started thinking less and less about a medical career and more about biodiversity, conservation of biodiversity, taxonomy of these little wasps, and the ants for a lesser part. And I kind of turned my back entirely on the medical field and decided I wanted to go into entomology. I didn't know exactly what, but I thought entomology was the thing for me. I happen to be good at it. I don't know why. I'm a collector by habit. That might have something to do with it. I'm good at organizing objects and figuring out things that are similar and different. It's almost like the career found me. And I just kind of... I had really bad grades up to that point, and then I got all straight A's. So once I kind of found this focus, and I've never looked back. I've been pursuing this now since 1995.
Great. Since graduation.
Yeah. Since then, and I went to a master's degree at Texas A&M, where I started on a parasitoid group that I still study today, the fidgeted wasps. Did my PhD back at Riverside. I moved back to Riverside because I loved the campus so much, and there was one particular professor I wanted to work with, and then I got a job with the federal government at the U.S. National Museum at the Smithsonian. So. Which, in my mind, is one of the finest institutions in the world for working on the taxonomy of parasitoid wasps. So,
Mike was talking to us that there's I think 35 million specimens in the collection.
Something like that. We're actually not sure how big the collection is. It's arguably the world's largest collection. It's a very historical collection. The founders of American entomology all have their specimens there. It's a living, breathing database of what we know about not only what's in the United States, but we have very strong holdings in the entire neotropical region, Mexico, Central America, and then the groups that I work on, I'm trying to build a world collection. So I've got now good samples from all throughout Southeast Asia, Madagascar, all of Africa, and large parts of Western Europe. So it's getting there.
Yeah, it's really great.
It's getting there.
And talking about biodiversity, the Hymenoptera group is huge, large, and have a lot of species. But And this week we saw that have a lot of groups that not even know what species there in some places. And what's the thing that all these species are around the world? Can you tell us about this huge biodiversity?
Yeah, sure, absolutely. So we have something like 115,000 named species of Hymenoptera. We estimate the true diversity is probably in the area around 3 million species. So we have a bit of work to do. And most of the large Hymenoptera, the stinging wasps and bees, ants, sawflies, those have all been largely described. There's still undescribed species for sure, but not to the extent that we deal with in the small parasitoid wasps, where we have, in the group that I work on, I would estimate we've named about 10% of the actual diversity. And I like that. I like the challenge of that. What concerns me, however, is we're losing expertise in how to handle these specimens, how to identify when something is new. These species have, not only are they obviously unknown to science, we don't have names for them, but they're providing ecosystem services through their biological attributes. We don't understand that. At the same time, while losing expertise, funding has more or less plateaued in the United States. So resources are limited. And on top of that, we're losing habitat very rapidly.
Sure.
And we're losing species before they can be documented. We're losing species that could be providing, and probably are providing, something essential for human life as we currently enjoy it. And that's where I take this job very seriously, and that's why I see my role, is trying to understand these species to the point where we can then do something about maybe keeping them around. We might decide certain species are very important if we like to eat corn without all kinds of bizarre genetic modifications or poison sprayed on them, etc. We might want to conserve species because they're maintaining a certain habitat that we as humans enjoy as on our weekends, etc. So I think we can all understand nature. We can all appreciate nature to a larger extent if we understand it a little bit more, that it's not this big unknown void. We might decide the conservation of a particular forest because it houses a unique lineage of wasp is very important, and maybe more important than a parking lot or a new high-rise. These are challenges not limited to certain parts of the developing world. These are the same challenges we face even in the United States and Western Europe, where we think we have a very sound concept of what biodiversity is in the United States, and for many insect groups we don't have a clue.
Yeah, and to solve this problem, the best thing is go to the field and collect.
You got to go out there and get it, because there is a large amount of specimens in the museum. We mentioned 36 million, just in the USNM alone, and it's very important to document what's already been collected, database it, understand it, identify it. However, the Earth is a dynamic system, and where things used to be collected may not be collected anymore. They might not be collected in the same numbers.
Sure.
And there's obviously lots of things out there that have not been collected yet. That's why it's very important to be collecting, and I think collecting responsibly, of course, not destructively. Conserving all the specimens, sending specimens to specialists, making sure specimens don't just sit around. They need to be utilized. And I think that is, without collections, we don't have, they are the data. That is the data. And if you don't go out and get it, it's not going to come to you.
Sure. You said to us, Tail, that some species, I don't think it's a genus or a subfamily.
Yep.
That in North America, you don't even have
the very enigmatic emarginine wasps, which are particularly diverse in Madagascar for unknown reasons, and are more or less devoid of any records being in the United States. However, a gentleman, an emeritus scientist in our museum, has been running a trap in his own backyard for something like 40 years. And right before coming on this trip to Argentina, I was sorting some samples for another project entirely. And there was an emarginine wasp in Annandale, Virginia. And it's not that it's not supposed to be there. It's clearly there, so it's supposed to be there. It's just a reflection of our ignorance. And it took 40 years of sampling in that one site to get one specimen.
Yeah.
There's a lot of rare things out there. And if you're not collecting and monitoring on a regular basis, these snapshots of biodiversity are going to be missed.
Sure.
Yep.
So we changed the place here.
Yeah.
Just to talk about huge thing that you told us in the course. That is the initiative of the Genome Project. Just tell to the people.
So the Global Genome Initiative, or GGI, is a Smithsonian project to essentially recollect planet Earth in terms of its biodiversity. But collect it in a very specific way by preserving the specimens to ultimately look at the genome, the complete genome of each species. So instead of a few pieces of the genome, which is what we currently do, we preserve things very carefully in very high-grade lab-level alcohol, for instance.
Just to make difference between species.
Yeah, or to build a family
around.
tree, something like that. That's very relatively easy to do. It's just a matter of doing some field work, sweeping around, or collecting things into the proper chemical, and you can get out little bits of DNA that you need. For doing a whole genome, the technology currently that allows us to do this requires specimens to be preserved perfectly while they're still alive. And perfectly, in this case, is preserving specimens into liquid nitrogen, freezing them instantaneously, ending all metabolic processes instantly, revealing tissues that are preserved more or less exactly as they are while they're in real life. Down the road, as the technology for genome sequencing improves, it may be found that this level of care was a bit overkill. Okay. However, we are working with the technology we have now, and it requires things to be frozen instantly. And using this type of experience here, coming to Argentina, collecting here, allows us to collect specimens for this project that can only be found here. This is essential. We talked earlier about the importance of field work. These species are not going to come all the way up through the Isthmus of Panama to the Smithsonian and present themselves. We have to go out and get them. It's a lot of work. It's a very different way of collecting. I'm teaching myself how to do it. We're figuring it out as we go along. The protocols are complicated. Specimens have to be databased a very specific way. Ultimately, however, what we're going to have is for not just humans and chickens and honeybee genomes. We're going to have genomes for arguably all the major lineages of living organisms on the planet, revealing in their genomes their secrets to how they operate, to how they navigate this very complex heterogeneous environment, how, in the case of parasitoids, they are able to fool their host into thinking there's nothing wrong with them, that this living being inside of them is supposed to be there when it's not, and it's seriously detrimental to their health. Right now, what we have is observational data on how these things operate that we as humans can look at. The Genome Project is going to give us an entirely different data set on what are the molecular underpinnings that are defining species and defining their behavior. What bizarre enzymes do they create? You name it. If we don't have the genome, we can't ask these questions. What we can find in that genome is similar, I would say, to what the Hubble Space Telescope has shown us about the universe. It's a different way of looking at the same data that's been in front of us as humans, sitting on planet Earth, looking up, seeing the Milky Way. Oh, that's interesting. The Hubble has allowed us to see into it. The Global Genome Project is going to allow us to see into nature on planet Earth. And what we can find in that is, I think, would probably be beyond our wildest dreams. There's no way we can anticipate what we're going to find. But the human endeavor to explore our own planet, to better understand our place in it, drives this. And I think it's one of the most exciting things I've ever been a part of.
Yeah, it's a huge challenge, isn't it?
It is a huge challenge, absolutely.
Because you can't use the specimens that you collected in these 20 years.
Nope.
And you can't use the insects and the collections around the world. So you have to get some collaborations.
Yep.
To make it possible, isn't it?
Yeah, this is not just an endeavor of the Smithsonian. This is an endeavor of the human race. That's what it's going to take. It's going to take collaborators around the world saying, "Hey, I've got this unusual thing. Can I send it to you?" And we're going to have to. Yeah, they can send it, but it has to be frozen. So that complicates things a little bit. But the nice thing about the Smithsonian is they've built a state-of-the-art facility for housing specimens that are sent in, and it's a public project. I mean, it's by public funds that are doing it. The data is going to be open to anyone in the world that needs the genomic data that we generate. The Smithsonian has a long history of having very solid funding, so that when things go in, they stay preserved the way they were anticipated. It's not something that some administrator could come along and say, "Hey, what is all this?" and pull the funding. It's safe from that. So we have that built-in security. So I feel, while it's very laborious, what I'm doing is a legacy, and I can now justify the legacy of the collection, the historical collection, because we're also adding to that while we're collecting for the GGI. But now we're adding to a new collection, a brand new collection, and that's very exciting. That's groundbreaking.
And it goes since insects and mammals, plants,
everything, microbes, you name it, deep sea trench worms, everything that we can get our hands on is going to go into this repository.
Yeah, it's a huge challenge.
Yep.
And have a lot of genus, species, families that are exclusive to some localities in the world.
And this is really where the historic collections are so important, because we can look at an animal, in this case a small parasitic wasp, in the collection that was maybe collected 70 or 80 years ago, and it tells us where and when we can find this thing. That bit of information is priceless, because now we know where to take all of our expensive, laborious gear to try to collect some new ones. If we didn't even have that, we wouldn't know where to begin.
Yeah.
So I see the GGI is simply a continuum of the National Insect Collection of the Smithsonian. It's just, you can't look at them. They're not on pins. They're in a giant vat of liquid nitrogen and a huge stainless steel casing. It's not quite as cool looking, I have to say, in my opinion, as pulling out a drawer of insects and looking at them all. However, like I said, we're after a different data set this time, and one that can have profound impact on natural science.
Matt, our public is made by biologists and biology students in Brazil and other countries, and I really like to know about you. If you can tell us, if you have to give them just one tip of if you had to choose one thing that you don't have to forget in your career, what it should be?
I would have to say the one thing I would recommend is always keep in mind that there's no question too small. There's no question so trivial it's not worth pursuing. And if you feel in your heart and you've looked at a lot of data, in my case it's a lot of specimens, and I see something that gets me interested, that's literally all I need to begin pursuing it in a more scientific way. That by observing the world and paying very close attention to it, questions present themselves. And I think the worst thing that an early scientist can do is consider that question is too small for me to spend some time with. I'm going to spend time on these bigger things. It's very important to pursue those small things because they become, invariably, they become the big things. You have to go into a situation completely naive. We have to go into the unknown, and you have to start somewhere. So don't consider any question too small. And if at all possible, please publish what you find. Because if you don't publish it, it stays with you. And that does a disservice to all of science. It needs to be out there published for the rest of the world to appreciate.
Great, Matt. Just to finish our talk here,
Yep.
I'd like to ask you if you have a internet site about the Genome Project or about the taxa group that you're working.
Yeah, interestingly enough, as I'm a federal employee, my ability to generate databases or public outreach websites is very strongly controlled by the federal government. So I don't have really fantastic websites I can send people to. If you Google my name, Matt Buffington, ARS, the Agricultural Research Service, that's the branch of the USDA I work for, you'll find all my papers there. You can download all those for free because they're federal government papers. If you Google the Global Genome Project at the Smithsonian, they do have a fantastic webpage. That's going to be the data portal for everything we collect. So lists of everything that we've collected are going to be populating databases there. So you can search for your favorite parasitoid wasp and see if we've gotten it yet. And if we don't have it, you can email us and say, hey, have you gone to this place? Because you're missing this very important lineage or something. So I would recommend people go there. If you're interested in Hymenoptera in general, hymencourse.org is your go-to place. That's the website we use for the Hymenoptera training course. And there are methods for preserving specimens, for making your own glue, for how to collect specimens. There's downloadable reprints of various very hard to get out of print material. That's a great portal right there. So eventually we'll have something a lot more profound, I think, as the GGI continues. I would like to give a shout out though to my good friend Simon Van Noort in Cape Town, South Africa. He has a website called waspweb.org.
Oh yeah.
And if you're into African Hymenoptera, that is one single resource that he's trying to aggregate all known records of Hymenoptera recorded in the Afrotropical region, how to identify them, updates in nomenclature, what their proper names should be. It's an amazing resource, and I think a model for what we need for North America, Central America, and South America.
Great.
Yep.
Matt, it was a huge pleasure to talk to you.
My pleasure.
We have to go out now and finish our work here.
Absolutely. We still have a lot to do.
It was a pleasure.
Thank you very much.
Valeu demais, pessoal. Até a próxima.